NASA wants to send humans on deep space exploration.
There's been a large call for exploration to Mars.
We cannot bring all of the food we need with us. We need to be able to grow it ourselves.
However, we don't know how the plants are going to respond in space.
And so this project is about figuring out how we can grow plants successfully and sustainably in the face of pathogens in space. I'm Anjali Iyer-Pacuzzi, and I'm a professor in Botany and Plant Pathology.
My name is Denise Caldwell. I'm a PhD candidate in the Department of Botany and Plant Pathology. In 2019, my professor was awarded a grant for a project working on defense responses in tomatoes.
Denise is one of my graduate students. She's been the one to figure out how do we grow tomato plants in this science carrier that NASA gave us.
We were going to look at plants that were grown in microgravity. What we wanted to look at was the defense responses.
NASA really needed to understand what happens to our food if we have a pathogen come through and completely devastates everything. One of the things that our experiment was designed to understand was, is plant immunity happening in a similar way in spaceflight as it is on Earth?
We elicited a response similar to a pathogen response here on Earth. We grew them, we froze them, and we brought them back down to Earth. And then we extracted the RNA and we sent that off for sequencing.
When we harvest plants for RNA extraction or transcriptomics, we want to understand their gene expression on Earth. We take a leaf, we put it into some liquid nitrogen, we freeze it instantly. Can't do that in space. And so we had to come up with a way to essentially flash freeze leaves, make them cold very quickly, and stay cold.
We sent it off so that they could read this RNA for us so we could understand what kind of responses the plants trigger when they're being attacked. The second part of that grant was to use simulated microgravity.
When you take away gravity, you are able to see connections, molecular connections between pathways that you might not be able to see on Earth when we do have gravity.
We wanted to see if there was any differences, number one, in the anatomy of these plants when they're growing in simulated microgravity, and if there's any differences in the way that the fungal pathogen colonizes the plants. So here on Earth, we know what pathogens do, but in microgravity or in zero gravity on the way to Mars, if we have a room growing full of vegetables and food for the astronauts, what happens if a pathogen gets in there? And so we need to know and understand how the plants are going to respond, because we don't want them to be 3 million miles away, stuck without any food.
One of the biggest things that comes out of it was that our plants grew really, really well in spaceflight, and just as well as they did on the ground. That was unexpected, partially because previous experiments have not shown that. And one of the reasons, though, is that plants haven't had all of the proper environmental parameters in previous experiments that they had for ours. Food is a huge part of everyone's lives, right? And not just food, but also plants in general, right? Helps with mental health, helps people remind themselves that in some ways that they're human.
The psychological effects for an astronaut to see a green, living plant and to be able to take part of that is part of who we are.
I think of this not just as nutrition and something that's going to sustain our bodies for years, but also something that's going to sustain, I guess I don't want to get too fancy, but to sustain culture, right? Because it's who we are as people. Taking care of plants will hopefully satisfy something within us.